Hi, I'm Archana Khurana from the lab of Dr. Mitch Kronenberg in the Department of Developmental Immunology at La Jolla Institute for Allergy and Immunology. Today, I'm going to show you how to prepare high-5 insect cells and infect them with baculovirus for the purpose of generating mouse CD1d tetramers. CD1d tetramers are used by our lab and by others for the recognition of NKT cells. Baculovirus systems are used widely to produce recombinant proteins including MHC class II tetramers, so the procedure you will be shown has much wider applicability. This procedure involves initiating and growing up high-5 insect cells, infect the cells with baculovirus carrying CD1d cDNA, harvest supernatant by centrifugation after 4 days of infection, dialysis at concentration of protein in 150 millimolar sodium phosphate buffer pH 7.4. Purification of mouse CD1d protein by nickel NTA agarose and ion exchange chromatography method. Concentration mouse CD1d protein to 1 mg per ml using YM30 concentrator. Enzymatic biotinylation of mouse CD1d by avidity protocol, elimination of free biotin by S200 column, and finally production of alpha-galactor CD1d tetramers. Though the whole procedure involves all these steps, but today we will only show you how to handle these cells and infect them with